So just as a quick intro, my name is Ramji Srinivasan, and I'm co-founder and CEO of Takeo. Our mission is to make immune measurement a reality for clinical trials. We're going to talk today about high-resolution spectral flow compared to conventional flow and try to answer a question of what are developers finding. So this is going to be a literature deep dive. And as background, the problem that we're going to tackle today is this one from translational scientists. And you can read here a translational scientist question, which is, if a scientist is interested only in T cells, as that's how the drug works, why would he or she do the extra work to analyze the rest of the immune state? So this is near verbatim from an immunotherapy developer, and I'm sure many people have asked a similar type of question. To attempt to answer this question, we're going to go through a quick literature tour, and we're going to cover 3 papers, all from the recent literature. The first one was on melanoma and healthy controls. The second was on pancreatic adenocarcinoma. The third one was on prostate cancer. And there are either pre-treatment, phase 2, phase 1. And so all of these links are in the slide deck and then will be accessible as well online in the materials. Let's take the first example. This is the one in melanoma, and we're going to do a very simplified study design. This is from Van Zelm et al. In 2023. So in this study, they had 15 melanoma patients and 16 healthy controls, and they ran them through several panels, including a 27-marker resting T cell panel and a 20-marker activated panel, both on a spectral flow instrument. These are the actual panels in question. So on the left-hand side, you can see the 27-marker resting panel, starting with Ki67 down to CD39. And on the right-hand side, you can see the 20-marker activated panel, starting from TNF-alpha all the way down to CD127. So what were the results? In short, they found a lot of exciting statistically significant findings, and several were ruled out. For example, an increase in activated gamma delta T cells in melanoma compared to controls. IL-17A and TNF-alpha were down in melanoma patients' gamma delta T cells. And then the expression of CD57 and EOMs were increased in melanoma patient gamma delta T cells. So those are the statistically significant findings. Now the question is, what if? What if a parallel set of researchers just ran an 8-marker truncated panel? So starting over here, CD3, CD4, CD19, viability dye, etc. The authors were kind enough to respond with what might be a truncated panel. They didn't actually run this experiment, but this is a thought experiment that we're running today. So what now we want to do is rewind time and see what happens. So let's zoom in a little bit more detail. In this 8-marker panel, let's compare to the gating scheme for the panel 1, which is a resting population definition. And you remember TCR gamma delta were cells of interest. That's highlighted over here. That's resolved by not just CD3, CD19, but a bunch of other cells, as we're going to see shortly. To get to this population, you need CD45, viability dye, CD3, CD19, and TCR gamma delta. So a minimum of 5 markers of this 8-marker panel is consumed in gating gamma delta T cells. Now you can probably tell where this is going, because as you just look at this table, a bunch of these markers are inaccessible if you only have 8 markers. That is, you can't capture the entirety of the tree. So let's run this experiment, which is applying the truncated panel with the 15 melanoma patients, 16 healthy controls. This was the original task, to run these 2 panels side by side. Instead, if we replace it by one 8-marker truncated panel, well, now everything changes. You can see a little bit of the gamma delta T cells in melanoma patients because, we can resolve that one population, but you miss a lot of the other findings. So for example, IL-17A, TNF-alpha, those are missed, expression of CD57 and EOMs. So you basically get a missed or incomplete understanding of the findings. So we're going to a high level of detail for that first paper. For the second and third paper, we're going to perform a similar exercise. So let's take the second paper. This is from Zhang et al. 2023. This is in PDAC, and they had 13 pancreatic adenocarcinoma patients, and they were comparing non-responders and responders, and they ran a 21 marker panel. So let's zoom into that marker panel. So you can see at the top, live and dead, all the way down to GLUT1 and some other markers, H3K2, ME3. So this is a 21 marker panel they ran. And then in this case, we just created a sample truncated panel. But before we get to that, let's look at the statistically significant findings. So in the original paper, they saw that clinical benefit responders were comparable to non-responders. VDAC, HK2, HK32ME were up in clinical benefit responders, and VDAC1 and CD8 positive T cells and non-responders were up. And then the last finding was baseline levels of soluble IDO, LAG3, and PD1 were higher in non-responders. So kind of visually laid out, you can see this on the left-hand side, clinical benefit responders and non-responders. The cells that were up were VDAC1, HK2, KS67, et cetera, on the left-hand side, and the non-responders, VDAC1, Cyto, LAG3, and PD1. So this is the kind of difference between the subsets. Now, let's do this thought experiment of what if we ran just a truncated panel. So again, CD3, CD8, PD1, HLA-DR. And the short version is we miss a lot of stuff. So on the left-hand side, the clinical benefit responders and non-responder differences are almost all missed. The T cell memory subsets were comparable between the two populations or really didn't show any statistical difference. You wouldn't be able to find that out. On the non-responders, things like PD1 would possibly be found because we have that marker there. But the rest of the findings would be very difficult to find with the same marker panel. So that's an example of what might be missed using the second piece of literature. So let's go to our third and last study, which is prostate cancer. And this is a phase 1, a simplified study design. So this is 14 prostate cancer patients before and after therapy, response versus non-response. And again, on a 25 marker panel. This was Priceman et al. In 2024. So for this design, they had a 25 marker panel starting at the top from CD3, CD4, all the way down to PDL1, CD95. And we're going to go through, which is they had 3 statistically significant findings and several more in the paper. CX3CR1 in peripheral blood CAR T cells. This correlated with the response to immunotherapy. Then fewer CX3CR1 positive T cells were in the product before infusion, as well as several other cell types like PD1, LAG3, other activation markers, and CAR positive and endogenous non-CAR T cells. Okay, so these are the statistically significant findings. Now let's shrink down the panel once more, CD3, CD4, PD1 viability, and run this 8-marker truncated panel. Again, this is a thought experiment. We didn't actually run this, but one would have missed 2 out of 3 of these findings. So the CXCR31 in peripheral blood CAR T cells correlated with response, the other cells that were observed in the product before infusion, and the state markers as well. That would have been partially found because we have PD1 as a marker. So hopefully this gives you a quick tour. There are several more like this in the literature, and you can see these. If you only use 8-marker conventional flow, on the left-hand side, you can see a conventional flow 8-marker, and then a potential representative spectral flow cytometry, which can cover up to 25 markers. You get a much higher and higher resolution, depending on the number of immune subtypes you capture. And we went through the literature and looked at different studies. For example, Barber et al., who found PD-L1 on CD8 T cells that would have been associated with clinical response, potential subsets associated with chemokines, as well as CD28 and CAR-positive T cells. So these are the kind of example studies that are out in the literature, and there are multiple others like this as well. So in sum, we set out to try to answer this question, which is the drug developer's question is, if a scientist is interested only in T cells, as that's the main modality, why would he or she do the extra work to analyze the rest of the immune state? And the glib, somewhat glib and funny answer is from another CSO who said this, which is the immune system consists of more than one cell type. So hopefully that gives you a quick tour. And if there are any other examples you think we should cover, send us your ideas. Feel free to DM, email, or put it in the comments. And with that, we will take any questions. Okay. The first question is on the underlying 8-marker panels, and did you run the experiments? So no, we didn't actually run the experiments. These were basically kind of in silico in the sense of we were looking for what we would have missed had we run these panels. But no, we didn't run the experiments themselves. We made up the panels by basically taking the biggest panel and shrinking it down to what we thought would be a realistic representation. Good question. Okay. Right. Well, if there are other questions that people have, feel free to email afterwards or send DMs. Happy to answer any. And you can always reach us at taco.bio. So thanks everybody for coming today, and look forward to supporting your trial if you're interested in working with us down the road. Talk to you soon.